Today we're speaking with Dr. Emrah Altindis. He is located at Boston College in Boston, and he received his BS degree from EJ University in Turkey. He completed a Master's in Science at the Middle East Technical University and also in Ankara, Turkey. He then moved to Italy to work at Novartis Vaccines and to start his PhD at Bologna University in Italy. During his PhD, he developed a novel vaccine discovery tool, Protectome, and identified new immunogenic proteins for two important human pathogens. Then in 2011, he came over here to the US. He joined John McAnallis lab at Harvard Medical School for his first postdoc, and he identified new virulence factors in Vibrio cholera. And then in 2014, he began a second training in Ronald Kahn's lab at Joslin to work on potential link between gut microbiome and diabetes, which is why he's here today to talk about that. And in the Kahn lab, he discovered the presence of bioactive viral hormones and characterized viral insulins for the first time. It's really fascinating. He started his laboratory at Boston College Biology Department in 2018, and his independent research program established through the NIDDK K01 Award, congrats, that has focused on the characterization of microbial insulin mimics and their potential role in type 1 diabetes. Two G. Harold and Lila Y. Mathers Foundation research grants and a JDRF grant awarded to him help fund the T1D and celiac disease gut microbiome projects in the laboratory. And he's also and always been passionate about the equity in STEM education and has been one of the founding members of the department's DEI committee, actively collaborating with specific Boston College programs, McNair Scholars, Gateway Scholars, et cetera, to create a really nurturing environment and research opportunities for the first generation or underrepresented minority students in his laboratory. Welcome, Emrah. Thank you so much for joining us. Yeah, and I'm really interested in your talk today, so dive right in.
Yeah, so thank you so much for this introduction, Monica, and thanks to everyone who are joining online. So I also see some friends' names here, so it is great. So if this is okay with you, I would like to share my screen so we can go over the paper. And if you have any questions, you can just jump in, ask these questions, so we can have an interactive conversation related to this paper.
Great.
So first, acknowledgements and the authors of this paper. This was a highly collaborative work between different institutions. So I started this project in Dr. Kahn's lab when I was a postdoc at Joslin Diabetes Center. And actually, this is based on previous studies that were published by Shiho Fujisaka and Siegfried Ussar, who is here. He's a group leader in Munich now. And also my friend and colleague, Marion Soto. So I will talk about the protocol, but the lab was already working on the effects of gut microbiome on host metabolism. And when I joined the lab, I wanted to use some different tools to ask different questions, but still trying to understand this link between the gut microbes and the host functions. So we also collaborated with a group in Netherlands. Professor Black was the person who was leading this group, and they sent us some human samples. I will show it in figure 8. And Dr. Kulkarni helped with the beta cell, his group helped with the beta cells. So a highly collaborative work, and Kiati Girdar in my lab led this study, especially the last experiments. And Qin Huang, again in my lab, completed several studies with also Marion Soto. So let's start with the first figure that is talking about the project. In the lab, when I first arrived, Shiho already established this model. So we have one group of mice on chow diet and 3 different groups of mice on high-fat diet. These are B6 mice. And we fed them with a high-fat diet for 10 weeks, but we also started to use 2 different antibiotics, metronidazole or Vancomycin. And metronidazole is an antibiotic that specifically targets anaerobic bacteria, and vancomycin is targeting gram-positive bacteria. So by using these different antibiotics, you are remodeling the gut microbiome in different ways. So what you are changing is their gut microbiome composition. All right. So as you see, at the end of this 10 weeks of high-fat diet treatment plus the antibiotics in these 2 groups, there was a significant difference in their body weight between the chow diet and the high-fat diet groups, but there was no effect of the antibiotics on the high-fat diet groups' body weights. So body weight was not affected. So usually people work on they collect the fecal samples and work on the DNA to determine the microbiome composition in these studies. Instead of working on the DNA, in this study, I collected the fecal samples, and using a quantitative proteomics approach, I looked for the proteins in the host proteome in fecal samples and determined how microbiome alters the host's fecal or intestinal proteome. So this is a PCA analysis, and as you see, this is the chow group, and these are the high-fat diet groups. One of the limitations of this approach is this is a 10-plex experiment, but because this is a quantitative proteomics experiment, you can just compare 10 different samples in one experiment. So this was the limitation at this stage. But as you see, there is a significant effect of the high-fat diet on the proteome composition, which is expected. But there was also some effect of the vancomycin and metronidazole treatment compared to the high-fat diet. This heat map shows the differences even better. Red is more abundant and blue is less abundant. Chow groups in the first three. Can you also see my mouse, Monica?
Yes, I can.
All right. So chow group is here, 3 high-fat diet groups, 2 vancomycin and 2 metronidazole groups. So this is the first cluster of the proteins that are decreased by high-fat diet. And these are the proteins that are increased by the high-fat diet. But if you look carefully, you will realize some groups that are altered by high-fat diet, but with metronidazole treatment, they are normalized. This is some of the proteins are here, and some of the proteins are right here that are the levels are normalized by the metronidazole treatment. So this was a surprising result. We also looked for the functions that are affected, and several functions were affected. I will not talk about this because our time is limited. Then we made these volcano plots that you see here, figures ABC, compared high-fat diet to chow, high-fat diet to high-fat plus antibiotic 1, metronidazole, or high-fat diet compared to the vancomycin. This is the log 2-fold change, and this is the p-value. And the ones that we are showing here are more than 4-fold change, and they statistically very significant. When we made this figure, we were quite surprised that the top proteins that you see here, amylase or elastase 3B or pancreatic lipase, these are all important exocrine proteins. So these are the proteins that are related to digestive effects of the pancreas. These are here you see the differences of each of these proteins. Amylase is significantly decreased by the high-fat diet in all 3 groups. Elastase, which is important for the protein digestion, increased by high-fat diet, but further increased by metronidazole. And lipase, which is not affected by high-fat diet, is altered by metronidazole and vancomycin treatment. So the only difference between these 2 groups is the gut microbiome composition. So we were able to verify our proteomics results by using Western blot. So we were able to verify that our proteomics results are real. But we also collected the pancreas from these animals. And using Western blot, we checked if we can also determine these alterations in the pancreas tissues. And as you see, there is a decrease with the high-fat diet, especially with the metronidazole, but this is normalized with vancomycin treatment, or elastase is increased by the vancomycin treatment, or lipase is increased by high-fat diet, but although these animals are still on high-fat diet, it is decreased by the different antibiotics treatments. So this was very surprising and unexpected. When we started this project, we didn't have a goal to work on the pancreas, but the results took us to this exocrine function related interesting findings. So we started our experiment one more time, this 10 weeks of high-fat diet treatment. And this time we collected the pancreas. As any diabetes researcher will comment or will know, in pancreas is function, not only the pancreas function, but also the size is altered in obesity, in type 1 diabetes and type 2 diabetes. Although there are some speculations about the mechanisms, how this is affected by obesity or diabetes, we still do not know the mechanism underlying this. Several groups previously showed in mouse models that when they are fed with high-fat diet, they have a larger pancreas. So this is shown by several groups. But in this study, we showed that by using antibiotics, we were able to normalize the pancreas weight to the chow levels. So please remember the first slide that there was no difference between the body weights of these animals, but the pancreas size is normalized here at the end of 10 weeks of antibiotics treatment. Then we asked, do we really need 10 weeks of antibiotic treatment? Or can we make these animals first obese and insulin resistant? And then what happens if we use the antibiotics in the last 2 weeks while they are still on high-fat diet? As you see, there is no difference in the body weights of the antibiotics groups and the high-fat diet group, but they are significantly heavier than compared to the chow group. And with this just 2 weeks of antibiotic treatment, we were also able to normalize the pancreas weight again. So then we asked another question. So let's put these animals on high-fat diet for 4 weeks. Then we will add antibiotics treatment for 3 weeks, and then we will remove antibiotics, and they will continue on the high-fat diet. What is the effect on pancreas size? And when we remove the antibiotics from this experiment, the pancreas size was larger, indicating that this challenge by antibiotics is needed to normalize the pancreas levels. So we did several experiments to determine if this is hyperplasia or hypertrophy. So if the cells are becoming larger, or do we have more cells? And at this point, after 10 weeks of treatment of antibiotics, the number of the total acinar cells, the cells that are the main 90% of the pancreas, approximately 85 to 90% of the pancreas is made of these acinar cells. And there was no difference in the number of acinar cells for this area, indicating that the cells were not larger, but they proliferated in a previous time. We didn't observe increased fat in the pancreas. Any questions? Or would you like me to continue?
Anybody?
Emrah, this is Jack. This is, first of all, really interesting stuff. I'm somebody who's really interested in this idea of pancreas size changes accompanying diabetes. But I know less about microbiome and antibiotics, but I'm curious that the antibiotics, have other effects than just changing the microbiome. And, that's why we take antibiotics. We're trying to do other things with them. So how are you, and maybe it's further along in the paper, but how are you verifying that these effects are mediated through the microbiome and it's not some other immunological sort of phenomena that's affecting pancreas size?
Yeah, I think this is a great scientific question that we needed to address, because some of the antibiotics might be metabolized, although we think that these are not, but still they might have some independent effects on the host. So to address your question, Jack, we started this experiment, which is right here. So in this experiment, the mice were on high-fat diet for 6 weeks in total. In the last 2 weeks, we treated them with antibiotics. So I showed you the results but of course we are repeating with another cohort. At the end of this experiment, we collected the cecum samples from these animals. Okay, and we transferred the cecum of these animals to germ-free mice that had never seen, have never been exposed to antibiotics. So so we transferred the chow mouse cecum to chow germ-free mice, high-fat diet to the high-fat diet treated germ-free mice, and then we tried to see if we are able to transfer the phenotype. We waited for 2 more weeks, and then we sacked the animals. And the first thing was to check the pancreas weight. So in this case, the alterations in the pancreas weight was quite significant in all groups. As you see, the microbiome coming from the metronidazole and vancomycin mice was able to normalize the pancreas level to the chow fat diet mice levels, and this was not the case for the high fat diet mice. I think that this is the experiment that addresses your question. These animals have never been exposed to any antibiotics. They are just taking the samples from the cecum. Cecum is very rich with different bacteria, and after 2 weeks, they had this effect, indicating that this is directly mediated by the gut microbes.
I gotcha. I guess I gave you the perfect segue there. But one other question: these mice were never fed a high-fat diet. They just had the cecum transfer. That's it.
No,
Gotcha.
so yeah, no, these animals, these 3 animals that you see with the pinkish color, are all high-fat diet before the cecum transfer, and this chow animal was on chow diet before the experiment. So they were already exposed to high-fat diet, but they didn't have any gut microbes. And we transferred the gut microbiome from metro to this animal, vanco to these animals, and high-fat diet to these animals. So yes, they were on high-fat diet for 4 weeks before the experiment.
I see. What about animals that are on a high-fat diet but that are germ-free?
Okay, we didn't add one control that was just germ-free to this experiment. Yeah, I think that it might be a very good control, but we don't have this one in this experiment. But very good question. So then the next question was, okay, we see these differences on the size of the microbiome, but is there any effect on the exocrine function? So we collected the pancreas of these animals, extracted the proteins, and ran Western blots. The sample size is 3, but I think the trends of the change are obvious. Like there is a trend of increase on the amylase in the vancomycin group, and so I can maybe use the quantifications here. So there's a trend of increase with the amylase group, vancomycin group in the amylase. No change in the lipase, but there's a significant increase on the high-fat diet as expected for the high-fat diet group, and there's a trend of further increase with the vancomycin microbiome.
Hi, Emrah, can I interrupt one question?
Please.
I think it's great that you see an effect from the fecal transplant, obviously. So the question I have here is that, is it really microbiome that you are transplanting, or is it a byproduct of the microbiome you are transplanting? So I'm asking you this question because there's a lot of publication, quite a number of years now, that's showing that the byproduct of the bugs, i.e. they are producing quite a number of short free fatty acids, which are highly beneficial, that potentially give you the same result here. At the same time, so at the same time, one other thing that I would like to add on is more like a comment, is that the antibiotic are known to also stimulating the GLP-1. And the GLP-1 may also lead to these beneficial effects as well. So the thing I'm not really clear here is that either the free fatty acids stimulate GLP-1 and GLP-1 that have these effects, or the free fatty acid, or the microbiome itself have the direct effects.
Yeah. So another excellent question. So one of the limitations of this study is that we didn't identify a direct causal bacterial species or strain or metabolite that is causing these alterations in pancreas. So, but previously, Shiho Fujisaka and Dr. Khan, my former mentor, Dr. Khan, and who is also a co-corresponding author in this study, they published a paper showing that by changing gut microbiome, you totally change the plasma metabolome and also fecal metabolome as well. So antibiotics and the gut microbiome and this crosstalk between host and the microbiome is altering all the plasma metabolome. So, but we do not know which metabolites might be related to these functions. But related to your point on GLP-1, the next experiment was I will show you in a minute. So here we show the effects on the exocrine function and pancreas size and also host fecal proteome. So the next question was, what about the endocrine functions? How does gut microbiome affect the endocrine functions? And as you said, some people already show that GLP-1 is altered by gut microbiome, so it is already reported. But in our study, in this figure, you see the results from the donor mice on the left panel, and the germ-free mice is always on the right panel, recipient animals. So, for instance, for GLP-1, high-fat diet caused a decrease in the circulating GLP-1 levels. And this was same for metronidazole, but vancomycin tended to increase, but this didn't reach statistical significance. But again, this is related to, it might be related to the sample size. But when we transferred the microbiome of these animals, the cecum of these animals to the germ-free mice, and after 2 weeks measuring the GLP-1 levels, this decrease was significant for high-fat diet. And then with vancomycin treatment, sorry, not vancomycin treatment, the gut microbiome transfer from vancomycin treated mice increased the GLP-1 levels significantly and normalized it. So this was a significant effect on this incretin. As some of I think as all of you know, GIP is another incretin that is very important for insulin sensitivity. And there was a trend of increase here with high-fat diet and a decrease with metro and vanco, but this was not significant. But in the germ-free mice, these differences were quite significant, and the gut microbiome was directly affected circulating GIP levels. CCK is an important hormone for exocrine function of pancreas for producing these digestive enzymes, and there was a significant decrease with vancomycin treatment, and this might be one of the reasons why they had smaller pancreas, because CCK stimulates proliferation of the acinar cells. PYY is another important endocrine hormone, and we identified these trends of decrease and increase, but all of these became significant in the germ-free mice, indicating that the gut microbes are not only affecting the pancreas size, not only pancreas exocrine functions, but also the endocrine functions of these animals. Is this?
Thank you. That's very cool.
Okay.
So,
yeah, please.
So, go on. I'm sorry.
No, yeah, please. Any questions are welcome. So, in the next slide, in the next figure, we will talk about the effects on the metabolism. Because, okay, these are the effects on the endocrine function. So what else? So previously, again, Shiho Fujisaka, Marion Soto, and Siegfried Usar in ConLab used this model to check the effects on the metabolism. But in this study, and one of the reviewers asked, okay, you are seeing these differences, but what about the chow-fed mice? Previously, we never used chow-fed control with vancomycin and metronidazole, so they wanted us to complete these experiments for this paper. So this is a GTT experiment, glucose tolerance test. We injected glucose, we intraperitoneally injected glucose to these animals, And this is what you expect. The blood glucose levels increase. And then in insulin sensitive animals, they go down. If the animal is insulin resistant, it goes down very slowly. As in the vancomycin and metronidazole treatment mice, they were more glucose tolerant. So you see a trend of decrease in the area under curve. And at the very beginning also, their blood glucose levels were much lower than the high-fat diet animal. But we were very surprised to get this result because these are animals that are on chow diet. They are already insulin sensitive, but by treating them with vancomycin or metronidazole, first their blood glucose levels on the basal conditions were, especially in the vancomycin group, was lower. But with the glucose tolerance test, this is the effect on the animals that are on the chow. But the vancomycin group was so glucose tolerant that the tissues just took the glucose. So this was a very surprising effect. It didn't increase, it just went down, and this is a specific effect of the vancomycin.
So quick question on that.
Please.
So on the child data, so obviously vancomycin definitely inducing insulin sensitivity. So, you know, do you check, is it associated with the insulin itself or its effects in the glucose or absorbed into the liver?
So yeah, is it affecting insulin tolerance or not? So to address your question, Gianluca, we completed the insulin tolerance tests in these animals to determine if they are more insulin sensitive or not. Because you asked this child question, I will directly go.
Oh, I'm sorry.
No. So I will just show the ITT results for this. So this is an insulin tolerance test. In these experiments, we inject animals with insulin. And in insulin sensitive animals, the blood glucose levels go down and then they start to come back, normalize, because insulin just affects. If there's any reader anyone watching who doesn't know the insulin function, because insulin stimulates glucose uptake to the tissues. As you see in these animals, again, in the vancomycin group, the blood glucose levels were lower. But in addition to this, in the vancomycin group, we were quite concerned that the animals were just about to become hypoglycemic. They were so insulin sensitive. And this was a surprise for us because we were not expecting to see an effect on the chow, in the chow animals to become even more insulin sensitive.
Oh, that's incredible. That's an incredible data. So how many weeks do you treat on the animal again?
So in these animals, these animals are the animals that are treated with antibiotics just for 2 weeks. This is the result coming from 2 weeks of antibiotics treatment.
No body weight change on this guy, right? Or food consumption change, right?
No, they didn't have a change in their body weights. Neither these animals nor these animals, the high-fat diet animals. But the result in the high-fat diet animals were also interesting. This is again ITT experiment, insulin tolerance test, and the vancomycin group, the green, and the blue is the metronidazole group. They were also significantly more insulin tolerant than the high-fat diet mice. So again, by altering gut microbiome, you can make these animals more insulin sensitive. This was already shown in the previous papers by my colleagues, and I am also co-author in some of them. But this was very surprising.
Yeah, it is indeed. So the insulin sensitivity here is that based on what you've done, or maybe you will show to the next slide, is it due to the gut incretin or due to the microbiome directly?
I think that it is quite complex. There are several factors. In a previous paper, we showed that there are several metabolites that are strongly associated with insulin sensitivity, but we do not know exactly which of these metabolites are responsible from this, but it looks like it is very complex. The increase in the GLP-1 levels or the effects on the PYY, GIP might be related with this insulin sensitivity. And the previous research also showed that the gut, by changing gut microbiome composition, you are also changing inflammation in different tissues. And by decreasing inflammation, you make tissues more insulin sensitive.
That's true. So you've seen quite a substantial increase, but this animal do not have any impact on the food consumption.
No.
Right. So what do you think that is? Why GLP-1 and in general or the incretin have a role in have a quite strong data supporting its effects in food intake as well as improving engaging GI tract transit that, but in your study you see that level, but it doesn't have any impact on food consumption and body weight. What do you think is going on here?
So we didn't check food consumptions. We checked the body weight change. Body weight change was not changing, but we didn't specifically look for the food intake.
Okay, I got you. Do you plan, since the animal have a very robust in insulin, glucose oxidation, do you plan to look at energy metabolism? Are these animals more active? I.e., they oxidize all their nutrient that they're consuming compared to control?
We are planning several follow-up studies. Yeah, and
Okay,
this might be one of the good suggestions for this. So to continue, and I will.
Emrah, sorry.
Siegfried, please.
Hi.
Nice to see you.
Great to see you.
Yes,
I would just have a quick question to this insulin sensitivity thing, is because to what extent is it driven by differences in basal glucose levels? If you would look at the area above the curve where you have baseline normalization, would you actually still see a decrease?
So the question is, if we normalize this to 100%, and so there was still a decrease, but it was not as significant as this one. Okay. The reason why we wanted to show this figure was to be able to show this difference in the basal conditions, which was quite significant. And these animals had really very low blood glucose levels.
No, exactly, because that's a little bit the question that I wanted to get to what extent it's really the glucose disposal or gluconeogenesis plus intestinal glucose absorption. And also, I don't know if you have any thoughts on this one, but so is it really the disposal into the skeletal muscle, or is it the liver plus the absorption from the intestine that drives this?
Yeah, but he didn't do oral glucose here, right? So he did IT. So the glucose, the glucose get into systemic, right?
Yeah.
So right. So the data is very encouraging. So the box check for the future, if I am in your shoe here, is that I'm gonna run the insulinemic euglycemic clamp to really nail this down to to really establish the bona fide insulin sensitivity, as well as capturing your tissues glucose disposal.
That's
great. I would just be curious to hear Emrah's answer.
Yes.
No.
This is a great question. So also, is it muscle, brain, and other tissue? We don't know which one. So so I don't know the answer of this question. To address this, we can do an in vivo glucose uptake experiment, which is quite challenging, or a clamp experiment. I believe that previous studies, Siegfried, you were also involved, show that all the tissues, including brain, becomes very insulin sensitive in these animals. So I believe that this is glucose uptake by different tissues, and muscle might be the first one because usually it consumes most. But I don't know the answer of this question, but this might be addressed in a follow-up study.
Great, thanks.
This has turned into quite a brainstorming for future experiments. It's great.
Which is great, yeah. Which is always great to have good colleagues asking these questions. So the reviewers also asked this question. Okay, what about the effects on the exocrine function when we feed these animals, the chow-fed mice animals, with vancomycin or metrodinose also? We show the effects on the endocrine function on germ-free mice animals. We show the metabolic effects. So with vancomycin treatment, we made these animals, chow-fed animals, more insulin sensitive and more glucose tolerant. And they asked, what about the exocrine function? So we ran these Western blots again to determine the abundance of different exocrine enzymes. And we showed that there was no difference in any of the chow animals, metro or vanco groups, indicating that the gut microbiome composition change in these animals is affecting insulin sensitivity, but it doesn't affect the amylase, lipase, and elastase levels. I'm talking about the chow-fed animals, but there was always a significant effect on elastase, chow to high-fat diet, or this is one of the markers of the pancreas function, and it is also decreased by high-fat diet as expected. So the last figure of this study was related to human data because, okay, we did all these experiments, but is it relevant to human insulin resistance or human patients? To address this, we collaborated with the Dutch group in Netherlands, and they previously published a paper in Cell Metabolism looking, investigating the effects of vancomycin on insulin-resistant obese men. So these are all men. We don't have female samples, another limitation. And what you see here is that these are the patients treated with placebo on the left of this Western blot on the left side. And these are the patients treated with vancomycin on the right side. And in each lane, in each two lanes, the first one is the sample collected before the experiment is started, and the second lane is after 1 week. So before the experiment, after 1 week. And on this side, this sample is collected before they started vancomycin treatment, and this one is coming from the samples after the treatment. All right. So we ran these Western blots for each of these amylase, elastase, trypsin, and lipase for 4 exocrine enzymes. And then we quantified these results based on placebo before and after, or vancomycin before and after. We were very happy to see that there was a significant effect of the vancomycin treatment in human insulin-resistant obese men. There was a 2-fold increase in the amylase levels with just 1 week vancomycin treatment. This was also the case for elastase treatment, 50% increase with the vancomycin treatment. But, and here you can see also the change per individual. But this was not the case for lipase or trypsin. So for these 2 enzymes, there was a significant effect of this vancomycin treatment in humans. So this was the final figure of the study. We believe that this is one of the first studies investigating the role of gut microbiome on pancreas function. And we show that it affects exocrine function, so these digestive enzymes, the pancreas size, but it also affects the endocrine function, the incretins as well. It makes these mice more insulin sensitive and glucose tolerant. And with the final figure, we were able to show that in insulin resistant obese patients, just changing gut microbiome, you can also change their exocrine function as well. So there are also some limitations of this study that we are aware of, but we will try to follow up, publish some follow-up studies in the future to address which specific metabolites or the species are responsible from insulin sensitivity or increased GLP-1, GIP incretins, and some other several. We have so many questions now.
Yeah, this is really, really fantastic. It opens the door to so many different experiments. I hope you have a long list of postdocs and graduate students ready.
Yeah.
And I just wanted, we went over a little bit. Are there any further questions for Emrah?
Hey, thanks so much, Dr. Emrah. And thank you for putting this together, Monica.
Yeah.
Are there any specific gut bacteria strains that are suspected to be helpful in limiting type 1 diabetes?
Type 1 diabetes. So, okay, in type, so gut microbiome composition of the type 1 diabetes patients are significantly different from the control healthy individuals. And people used NOD mice, the main mouse model that we use in type 1 diabetes field, to determine the role of some of the bacteria. And they show that, for instance, Akkermansia municipale, the most famous bacterium, I think, in the probiotics field, Akkermansia, was protective for NOD mice. Then SFP, another. So they show 2 or 3 different protective ones. We have an ongoing study. We show that Parabacteroides dystosonis, this paper is now under revision in PNAS. We show that one bacterium is increasing the type 1 diabetes rates. So new studies, we need more studies to determine the causal links, but the gut microbiome composition is totally different in type 1 diabetes patients, also in type 2 diabetes patients compared to healthy controls.
Yeah,
but
the diversity decreases dramatically at time of onset in human type 1 diabetes patients, correct?
Yes, so they are less diverse, and the gut dysbiosis happens. The gut becomes more leaky, inflammation is increased, the regulatory cells are decreasing, also in the intestines. But then in all microbiome studies, this question comes: is it chicken or egg? Which one comes first?
Yeah.
Is it causal of the disease, or does disease cause this? Germ-free mice experiments are very helpful to address these questions.
Yeah, and the body of literature is expanding, right? So it's very interesting.
Yes,
So a
yes.
company like Finch or Holobiome or something could be very instrumental in kind of, using their muscle to further this field with collaborations with scientists. So
Right.
yeah, it's
Right.
really, really interesting times.
Yes, so many interesting times, and we need more causal studies. So yeah, because we have now enough, we have more than enough associative studies. We show that there are these associations, but now we need to go deeper to identify, okay, what, for instance, for my study that I just talked about, okay, what is the bacterial species that makes us more insulin sensitive, or what is the metabolite? So this is the follow-up study for this one, for the pancreas study.
Okay, super. Thank you so much.
Yeah, thank you.
Thanks. Well, maybe you'll have some more conversations offline. I hope so.
Okay.
Okay, well, thank you all again. We've gone a little bit past. Thank you, Emrah, for this really, really interesting talk, and for everyone's comments. Totally appreciate it. And thanks again for participating in Ask the Expert, and have a great rest of your day.
Thanks so much, Monica.
Bye-bye.
Thanks, everyone. Bye.